Good afternoon. It's a great pleasure to welcome everybody to our CRC 1182 seminar series. And it's a great pleasure to welcome Bob Rosier. The plan was very different. The plan was that Bob Rosier should arrive last night in Hamburg airport from Valencia, and then should be here and should visit labs and talk to people, give a seminar, talk to me, and we go out with a number of students then for a dinner. Unfortunately, the current situation doesn't allow us to do that, and so on Friday we decided to cancel the personal visit of Bob, and so we are back here in our online format, which is okay, and I'm very thankful to the technical help in the background who makes that so nice possible. Bob will give a seminar now, and he's very happy to take questions after the seminar. Please ask your questions directly by raising your voice. You can also use the chat room, but it's easier, and we are not so many. We are between 40 and 50 people, that you just get your voice in the meeting here. So Bob Rosier is, I would call, it's not an official status, but he's a senior PhD student in Human Microbiome Laboratory by Alejandro Mira in the FISABIO Foundation in Valencia, Spain. That is co-financed by the European Union, a research structure which is very well organized and also equipped. And Bob got his early training in university training at the University of Amsterdam and also in Eindhoven. You may know Eindhoven is the Philips laboratory, and so he spent some time there. And then from Amsterdam, actually he went to Germany in the Max Planck Institute of Terrestrial Microbiology in Marburg to study what we call, hardcore symbiosis in a cockroach, in a gut microbiome context with Professor Brune. But as he wrote us in the forefield before we arranged this visit here, he wrote that after 4 months of being in the lab working on cockroach gut microbiota, I cite him now, I discovered and I wanted to return to my real passion, which is the human oral microbiome. And here we have one of the real experts in the human oral microbiome. I met Bob in the Gulbenkian Summer School some time ago, where he also presented already his work. He's a fantastic junior investigator, has more than 10 papers, 2 scientific report papers in the meantime, and is very productive, and is now eager to share with us his new findings, and we are very lucky and very happy to have him. And I'm sure you will now learn all kind of different unexpected insights, what happens in your oral cavity with your microbiome in context with the diet, in context with the physical activity. Bob, the stage is yours. I don't have to read the title of your colloquium anymore. It's from improved sport performance to dental caries prevention, and Bob actually discovered the role of nitrate which is in the context of the oral microbiome. The stage is yours. Very happy to listen to your talk.
Thank you very much for this kind introduction, Thomas. Yeah, it's indeed a shame we had to change our plans. I was looking forward very much to my first trip abroad professionally after the start of the pandemic, but unfortunately the current situation did not allow it. So we will keep that for the future. So I will talk about the benefits of nitrate reduction by the oral microbiota. I am very happy that I can still share some of our results with you at the Meta-Organisms Research Center. And the benefits of nitrate reduction by oral bacteria range from improved sport performance to dental caries prevention. We have been focusing mainly on the oral effects, but I will also introduce some of the systemic effects that include sport performance. And the reason that you see some leafy greens and beetroots over here is because they are vegetables that are particularly high in nitrate, and the benefits that I will mention today can be stimulated by the consumption of those vegetables. So first of all, I will introduce the oral microbiota, the human oral microbiota. You may not have heard as much about this. We usually focus on the gut microbiota, but the oral microbiota is also important for our health and even contributes to our physiology. And this community is diverse and stable. Each individual contains between 100 to 200 or more bacterial species of the 700 oral species that have been identified. The predominant bacterial phyla are Firmicutes, Proteobacteria, Actinobacteria, Bacteroidetes, and Fusobacteria. And there are also archaea, protozoa, viruses, and fungi, but these remain less explored. So these microorganisms form biofilms on all oral surfaces. Different habitats are the tongue, the teeth, and the gingival crevice, which is the space between the teeth and the gums, and the buccal mucosa, which is the interior of the cheek. And the biofilms on the tooth are called dental plaque, and they are associated with the most common oral diseases, namely caries and periodontal diseases. So over time, these biofilms grow and accumulate, and we have to apply oral hygiene to start with a clean tooth surface again, and then the biofilm formation starts again. Bacteria from all these surfaces shed into the saliva that leads to high amounts of bacteria per milliliter of saliva. It's probably more than you expect, but we have between 10 to the power of 7 to 10 to the power of 8 bacteria per milliliter of saliva. And before I want to explain oral disease development, I would like to go over the concept of dysbiosis and compare it to disease by an external pathogen. So this is a very simplified representation of a healthy oral microbiota on a human body surface. This could be the skin or the mouth. And when an external pathogen arrives and is able to colonize, it can cause disease. And sometimes we have to take antibiotics to remove it. In the case of dysbiosis, you start with the same healthy oral microbiota or any microbiota, and due to bad habits like an unhealthy diet or inappropriate hygiene, some species overgrow, and this orange bacterium over here, normally it's innocent, but when it increases in number, it can cause disease. And this increase in disease-associated species and functions is what we call dysbiosis. And we find dysbiosis in all oral diseases, in caries, in periodontitis, which is a severe form of gum disease, and in halitosis, which usually results from dysbiosis of the tongue microbiota, and it's a fancy word for bad breath. So these oral diseases can start with certain stress factors, and I will explain the development of caries and periodontal diseases briefly. In the case of caries, the stress factor is carbohydrate consumption, especially sugar consumption, and our microbiota ferments these carbohydrates into organic acids, of which the most important one is lactic acid. This leads to a pH drop, and a low pH and the presence of carbohydrate select for acid-tolerant and saccharolytic species that are better at fermenting carbohydrates, and a vicious circle is formed that's until the pH levels drop at levels in which enamel is demineralized, and these nasty cavities can form over time. That can also be very painful. In caries, we see an increase of bacteria belonging to the phylum Firmicutes. In the case of periodontal diseases, the main stress factor is usually a lack of oral hygiene. Because of that, dental plaque accumulates, and our body responds with gingival inflammation. Gingival inflammation includes more leakage out of the space between the teeth and the gingiva of a protein-rich serum. And the presence of protein and inflammation select for inflammation-tolerant and proteolytic species that further accumulate, and a vicious circle is formed. The initial stages of gingival inflammation are reversible, so if you remove the biofilm, the inflammation will go away. But long or repeated episodes of gingivitis can lead to a chronic and destructive inflammation that's called periodontitis. And in periodontitis, host tissue is lost and teeth can fall out. And we see that bacteria from the phyla Bacteroidetes, Fusobacteria, and Spirochetes increase. Although these types of dysbiosis are different, they share something in common. In both diseases, representatives of these genera, Rothia and Neisseria, decrease. And these are both nitrate-reducing bacteria. And I will get back to them later in my talk, so it would be good if you could remember them. And that was actually the first step that got us interested in nitrate reduction, the observation that these bacteria were always decreasing in disease. But I will come back to that later. The impact of the oral microbiota is not limited to the oral cavity, because the oral cavity is part of the gastrointestinal tract and the respiratory system. It's also directly connected to the bloodstream via the highly vascularized oral mucosa. And products of periodontitis, for example, products of this inflammation can reach different parts of the body, cytokines, bacterial products, and bacteria themselves. And periodontitis is strongly associated with many diseases, including cardiovascular diseases, pregnancy complications, liver problems, and gut dysbiosis and gut disease. And some causalities have been found. I will show you one in the next slides of a recent study where they found the bacterium Porphyromonas gingivalis, which is the bacterium with the strongest association with periodontitis in Alzheimer's disease brains. And they found the toxic proteases of this bacterium as well that correlated with the pathology of the disease. So the more proteases, the worse the state of the patient. So the authors infected mice with this bacterium, and they saw that it managed to colonize the brain and increase amyloid plaque protein formation. And amyloid plaques are the black dots that we find in the brains of people with Alzheimer's disease that also correlate with the pathology. So the conclusion of the authors was that in periodontitis, P. gingivalis could enter the bloodstream and over the years colonize the brain and contribute to the development of Alzheimer's disease. And they also saw that with small molecule inhibitors that block these toxic proteases, the mice were more healthy. And this is very interesting, but also a bit scary. But do not worry, because the oral microbiota also does many good things. We need it in health to prevent the colonization of foreign pathogens. And it also has an essential part in the nitrate, nitrite, nitric oxide pathway. This pathway starts with the salivary glands concentrating plasma nitrate into the saliva. When we're fasting, we have between 100 to 500 micromolar of nitrate in the saliva, which is already 10 times the plasma concentration. And after a nitrate-containing meal, this goes up to 5 to 8 millimolar. And foods with nitrate include many fruits and vegetables. It's particularly high in leafy greens, such as spinach and lettuce and cabbage, and also in beetroots. And oral bacteria reduce this nitrate to nitrite, and nitrite is converted into nitric oxide in our body. Nitric oxide is involved in a broad range of processes, and one of them is vasodilation. Nitric oxide is an important vasodilator of the human body. And over here on the right side, we have the classical pathway that our body uses to produce nitric oxide. L-arginine is converted into nitric oxide by our enzymes called nitric oxide synthesis. And on the left side, we have the more recently discovered pathway in which nitrate from the diet is reduced to nitrite by oral bacteria. And while our body cannot reduce nitrate to nitrite efficiently, we have several enzymatic and non-enzymatic processes that reduce nitrites to nitric oxide. And this nitric oxide can also oxidize back to nitrate. That's why we also have nitrate in our saliva when we're fasting. And what they found in 2013, they got firm evidence that when you sterilize the oral cavity with chlorhexidine, there's no more nitrite produced by oral bacteria, and the levels of nitric oxide drop and blood pressure increases. And I will explain a little bit better how this goes in my next slide. So here's an individual taking a glass of beetroot juice full of nitrate. And when we swallow this beetroot juice, like other nutrients, the nitrate gets into our bloodstream. And when the blood passes by the salivary glands actively concentrate this nitrate into the saliva so that oral bacteria can reduce the nitrate into nitrite. We then swallow the nitrite produced by bacteria. That gets into our bloodstream again. There it reacts with hemoglobin, forming nitric oxide. And nitric oxide is a vasodilator, meaning that it leads to an expansion of our blood vessels. And if you use very strong mouthwash with chlorhexidine or even regular mouthwash on a regular basis, you could kill too many oral bacteria and lose the production of nitrite. And then the nitric oxide that was produced from that is also lost. And this part of vasodilation is also lost, which increases blood pressure. So we want to avoid that. And you probably shouldn't use mouthwash if you're healthy. And if your dentist doesn't recommend it, to keep this pathway intact and functioning well. And not only because of blood pressure, because it has more benefits in the body. They were summarized in this review published in Cell Metabolism in 2018. And it states the described cardiometabolic effects of dietary nitrate from experimental and clinical studies include the lowering of blood pressure, improved endothelial function, increased exercise performance, and reversal of metabolic syndrome, as well as anti-diabetic effects. And to obtain these effects, you need a process involving seemingly symbiotic oral bacteria that I just showed to you. And I won't explain all of these cardiometabolic effects, but I will show you a little bit about sport performance. I feel like I'm obligated to do that as it was in my title. So here's a review about dietary nitrate and physical performance, also from 2018, by Andrew M. Jones, who is probably the big expert about nitrate and sport performance. And what they summarize is that nitrate in the form of nitrate salts or vegetable juice, usually beetroot juice, increases nitric oxide in plasma, improves the efficiency of oxygen usage in the liver, and increases sport performance. So here are examples from moderately trained individuals in different studies, cycling or running. And here the significant difference in the presence of nitrates after nitrate consumption is indicated. And whenever it's significant, there was an increase in sport performance between 0.5 and 2.5%. And that is, of course, interesting if you're competing. And this is all thanks to the activity of oral bacteria. Some of the potential mechanisms are described in this review, and nitric oxide appears to improve the oxygen delivery to skeletal muscles, also reduce the oxygen cost of mitochondrial ATP resynthesis to produce ATP, and also reduce the ATP cost of cross bridge formation, which is involved in muscle contraction. And altogether, these mechanisms reduce the oxygen cost of exercise and improve exercise performance. So yeah, I found it very fascinating that the bacteria in our mouth are affecting sport performance. But we are in the oral microbiome lab, so our interest was mainly, or our focus was mainly on oral processes, what could be going on inside the oral cavity, and could this be important for oral health? And when we started this project, there were several papers out there from which we could get some hypotheses. Some were focusing on other microbial communities like terrestrial or in water. And oral bacteria can reduce nitrite further to nitric oxide. Nitric oxide, apart from having these cardiometabolic effects, is also an antimicrobial molecule, so it could kill sensitive species. Alternatively, nitrite can be reduced to ammonium, which is the ion of ammonia, And during these reduction processes, protons are used. So that could limit acidification. And acidification can lead to caries, so that could be beneficial for dental health. And finally, we noticed that nitrate-reducing bacteria can use lactic acid and convert it into weaker acids. And when this is done, electrons are released that are used in parallel, in this reduction process. So lactic acid usage happens in parallel with these reduction processes, and that could further limit acidification. So based on our hypothesis, we set up a study to see if nitrate would affect the oral microbiota in vitro. And in a 96 well plate model, we grew saliva of 12 healthy donors in sugar-rich medium with or without 6.5 millimolar nitrate. And after 5 hours and 9 hours of growth, we saw interesting things. The nitrate was used up and there was nitrite produced. So here is the nitrate in milligrams per liter in the spent medium. And this is 0 hours, the nitrate that we added. In the control condition, there's also some nitrate that was in the saliva, but very little. This is the nitrate that we added. And after 5 hours, most nitrate had been reduced. And after 9 hours, there was no more nitrate detectable. Nitrite first increased and then seemed to have been metabolized as well. Ammonium increased after 5 hours in both conditions and further after 9 hours, but it was significantly higher in the nitrate condition at both time points. Then lactate, the ion of lactic acid, first increased and then decreased, but was significantly lower at both time points in the nitrate condition. And in accordance with these reduction processes happening and less lactate being detected in the nitrate condition, the pH was significantly higher in the nitrate condition at both time points. And actually, this pH drop over here in the control condition was significant, but here it was only a trend. So nitrate clearly was preventing acidification, which is very interesting for oral health. And we were very happy with these results, but it became more interesting when we looked at the bacterial composition. Here are the averages after 5 hours and 9 hours. The inner circle is the nitrate condition and the outer circle is the control condition without nitrate. And I won't go over all bacteria here. I have a better figure in my next slide for that. But one of the things I do want to show you here is this yellow bar, that's Neisseria, increases consistently in all donors in the presence of nitrate. So that was one of the significant changes that we observed. And I will show the rest of the significant changes here in this graph. This is the log2 of the nitrates divided by the control, relative abundance. And whatever is up here was higher in the nitrate condition. Whatever is down here was lower in the nitrate condition. And we saw that Neisseria increased and also Rothia. And Rothia and Neisseria are both health-associated and nitrate-reducing genera. Then if we look at the rest of the changes, these genera are very interested because all of them are either associated with one disease or another. And we could nicely cluster all significant changes in health-associated. Those were the bacteria that increased. Caries-associated, Streptococcus and Veillonella are firmicutes that are strongly associated with caries. Oribacterium is also associated with caries. And the other genera that decreased were associated with periodontitis and halitosis. So it really seemed that nitrate was decreasing dysbiosis or increasing eubiosis, which is an increase in health-associated species and functions. And I summarized our results and the results of other studies in this figure. So when we consume vegetables with nitrate, there's a direct effect. When we chew on them, then the nitrate in our saliva increases, and there's an indirect effect when we swallow them because the plasma nitrate is concentrated by the salivary glands into the saliva. They have these special transporters for it. And nitrate is reduced to nitrite by oral bacteria. Part of it is ingested, which leads to the systemic effects. And nitrite is also further reduced to nitric oxide by oral bacteria. This also happens at a low pH, and this pH is found in caries sometimes, so that could give negative feedback to caries. And since nitric oxide is an antimicrobial molecule, it can lead to changes in the biofilm composition. These anaerobic genera associated with halitosis and periodontal diseases are sensitive against oxidative stress of nitric oxide. So we think it's likely that that's a possible mechanism that could explain their decrease. And we also see that the biofilm changes because nitrate reducers have a selective advantage. They like nitrate, they use it in their respiration. Denitrification is bacterial respiration. And nitrite can also be reduced to ammonium, the ion of ammonia, and these reduction processes, as well as lactic acid usage, increase the pH of dental plaque, which can prevent caries development, and it can also limit the overgrowth of these bacteria that like a low pH. They grow well at a low pH, and when you buffer the pH, you prevent their overgrowth. And finally, this little part over here, we added that based on sewage microbiology, because when sewage smells bad, they actually throw in nitrate to reduce the bad smell. So we looked into those mechanisms, and it's because denitrification is more energy efficient than sulfate reduction. So as long as denitrification can take place, sulfate reduction is inhibited, and the reduction of sulfates leads to hydrogen sulfide production. And this is a stinky breath. It's one of the main gases involved in bad breath. So we think that nitrate must also be limiting that inside the oral cavity. And in the end, I will show you some data that supports this idea. And hydrogen sulfide can also be used as an electron donor during denitrification. So this process could really limit bad breath as well. Finally, apart from eating vegetables with nitrate, we think that oral care products with nitrate could be beneficial. And that's actually the next project that we plan that we will start in January. So most of these oral mechanisms were based on in vitro studies, on in vitro results. So we also asked the question, does nitrate work in vivo? And this year we published some first clinical experiments in this paper called "A single dose of nitrate increases resilience against acidification derived from sugar fermentation by the oral microbiome." And to prove this, we set up 3 clinical experiments that all supported the title, but I will only show you the results of experiment 1 and experiment 3 today. In experiment 1, we compared an active supplement with a placebo supplement. 12 individuals took them, both each on a different day. And at baseline, before supplement intake, they did a sugar rinse, and also 2 hours after supplement intake. The active supplement was a beetroot supplement, by the way, so it was full of nitrate, and the placebo supplement did not have any nitrate. Then in experiment 3, in 6 individuals, we compared a beetroot supplement with water. And again, individuals rinsed their mouth with sugar at baseline and 4 hours after active supplement or water intake. And I will explain why we did sugar rinses. So in sugar rinses, you take a pre-sugar saliva sample and a post-sugar saliva sample, actually a 10-minute post-sugar saliva sample. And the reason that we do that is because when we rinse our mouth with sugar, or when we consume candy or other sugar-rich things, there's a direct pH drop, which generally reaches its lowest point after 10 minutes. So by comparing the pre-sugar pH and the 10-minute post-sugar pH, you get information about the karyogenic capacity of the oral microbiota. And we did this under influence of nitric-rich supplements, and these are the results. First, we looked at the salivary nitrate to see if it really increased after supplement consumption. Here is the active supplement day, baseline, and 2 hours after supplement intake. And this is the placebo supplement day at baseline and 2 hours after supplement intake. And the nitrate-rich supplement increased the salivary nitrate 2 hours after consumption, while the placebo did not. And we saw a similar pattern during the other experiments. While the active nitrate-rich supplement increased the salivary nitrate, water did not. So that was expected, of course, but it had to be checked. And then we looked at lactate, the ion of lactic acid, and after every sugar rinse, lactate increased. What was interesting is that the pre-sugar pH was lower significantly 2 hours after supplement intake, but not 2 hours after placebo intake. And there was also a trend of the lactate being lower after sugar when individuals took the nitrate supplements compared to the placebo supplement. And this difference was significant when comparing the nitrate supplement with water. So in this experiment, the lactate production seemed to have been lower when individuals took the beetroot supplements. And finally, we measured the salivary pH that dropped after every sugar rinse, but it dropped significantly less when taking the nitrate supplement compared to the placebo supplement. And also when comparing the pH drops on the supplement day and on the water day, the nitrate-rich supplements seem to have pH buffering effects. And we found this very interesting because it's a single dose. And in the future, we want to see what happens if individuals take beetroot juice for a week or a month, because we think that the nitrate-reducing bacteria would increase and you would see stronger effects. But it's very interesting that beetroot juice, which has sugar itself, it has quite a lot of sugar, and still it's preventing pH drops because of its nitrate content. So that was quite interesting. Here I have some correlations from this study. So we found a clear negative correlations between lactate production and the pH after sugar. The more lactate there was produced after sugar, the bigger the pH drop, which supports the idea that this is the main acid, lactic acid is the main acid involved in these pH drops. And then we looked at salivary nitrate. We saw a positive correlation with the pH after sugar and a negative correlation with the lactate after sugar. So the more nitrate the individuals had accumulated in their saliva, the less lactate and the smaller the pH drop. That's what these correlations indicated. We also looked at the bacterial composition of experiment 3, which is of course a small group of individuals. So this is only a preliminary result, but it was very interesting. We looked at the relative abundance using 16S rRNA gene Illumina sequencing. And here is the baseline of the supplement day and 4 hours after supplement intake, and baseline and 4 hours after water intake. And there was one genus that increased significantly, and this was Rothia. And that's a nitrate-reducing genus I showed you previously. And we confirmed this small but significant increase by qPCR. And it was also really interesting to see that Neisseria and Rothia, the genera that were increasing in the in vitro study, correlated negatively with the lactate produced after sugar rinsing. So the more of these bacteria the individuals had, the less lactate was produced. And Neisseria also correlated positively with the pH after sucrose. So the more Neisseria there was, the less the pH dropped. And this is preliminary evidence that these bacteria could indeed be involved in nitrate-mediated pH buffering inside the oral cavity. And based on these results, we came up with the next research question that was, could nitrate-reducing species be used as probiotics? And to start this project, we first had to get nitrate-reducing species. So we started with dental plaque and tongue coating of very healthy individuals, adults without any oral diseases, with perfect teeth and gums, and a very healthy blood pressure. And what we did is we diluted their sample and we grew it on agar plates for 48 hours. And we were interested in the plates that had single colonies, so we could select the nitrate-reducing bacteria. And I will explain this method a bit better to you. It looks like this. So this is an oral sample on a plate, a lot of different sizes and shapes and colors of colonies. This is after 48 hours of growth. And then when we apply our staining method that I will explain to you in the next slide, you see that some colonies have a red circle around them, and these colonies are nitrate reducers. So how does this work? Here is a plate, but this time it's a side view. So you have the agar here with the colonies after 48 hours. And what we do is that we add one layer on top of it with agar containing 1 millimolar nitrate. So the nitrate reducing colonies reduce this nitrate into nitrite. And then we add another layer on top of this containing the well-known grease reagent, which forms a red color with nitrite. And only the bacteria that have reduced nitrate now have a red circle around them. So you can nicely isolate them with a sterile toothpick. You just go through the two layers and you take your colony to a new plate. You wait 48 hours so you get growth again, and then you take a single colony to another plate just to make sure you have a pure culture and not a combination of two bacteria, which can happen sometimes. This is just to increase your chance of having a pure isolate, and then you stain that again using the same overlay methods to confirm that it's really reducing nitrates. And this is how it looks in real life. So here are two nitrate-reducing isolates on a plate after staining. And we did this 62 times to obtain 62 nitrate-reducing isolates. So here we start with bacterial communities from the teeth and the tongue, and we got these 62 isolates. And we then did in vitro testing in liquid medium to get 6 probiotic candidates. And one of the tests we did was just see how much nitrate they reduced. So we added 6.5 millimolar nitrate, which is a concentration that you can find in saliva after a nitrate-rich meal. And we looked at how much of the nitrate they reduced. And we said, okay, we only want to work with species that reduce more than 20% of nitrate after 4 hours. And after 7 hours, they should have reduced 100% of the nitrate. So that was one of our selection criteria, to have good nitrate reducers. And other requirements for the selection of the probiotic candidates were no virulence genes. So we sequenced their genomes, and I will show you some genes in the next slide. We also wanted good aerobic growth. This is very useful for industrial purposes. If they grow well with oxygen, that's a huge advantage. And we didn't want them to produce acid, obviously, because that would cause caries, and they had to be health-associated species, but that was very easy because all 62 isolates were health-associated. And all these criteria led to the selection of 6 probiotic candidates. Here is a list of them: 1 Rothia aerea strain and 5 Rothia mucilaginosa strains. Here is their database number. You can get them in the Spanish culture collection. And we also added our best Rothia dentocariosa strain, just to have another Rothia species there. And what's interesting is that all 3 of those Rothia species have been classically associated with oral disease-free individuals. And we hypothesize that this is because of their ability to reduce nitrates. We looked at the genomes of these 3 Rothia species, and we looked at the genomes of our isolates of those 3 Rothia species, and we saw that they all had these genes that are in blue, which were related to nitrate and nitrite metabolism. So they could all reduce, they all had the genetic potential to reduce nitrate to nitrite, and nitrite further to ammonium, but also further to nitric oxide. They also had molybdenum binding genes, and molybdenum is an essential cofactor for the enzymes that reduce nitrate, so that also makes sense. We found lactic acid utilization genes in all strains, and we also found that there were no antimicrobial resistance genes or mobile genetic elements or virulence genes. And this is very important for any probiotic. They cannot have genes that could potentially harm us. And yeah, that's actually established by the World Health Organizations, and there are databases to check for these genes. And here are 3 typical colonies of these different Rothia species: Rothia aerea, Rothia dentocariosa, and Rothia mucilaginosa. And yeah, we wanted to see their effect on oral communities of our 6 probiotic candidates. So we used the same in vitro model as that I showed to you earlier, but this time, apart from adding nitrate, we also added 6 different Rothia isolates. And after 7 hours of growth with the isolates, we saw that more nitrate was reduced. So here is the nitrate at 0 hours. This is what we added basically. And then without an isolate, the oral communities reduce part of the nitrates. But when you add any of the 6 isolates, significantly more nitrate is reduced. Then there was also more lactate consumed by the isolates in combination with nitrate. So this is the lactate that we begin with that increases because the sugar in the medium is fermented by oral communities over time. And when adding the isolates in combination with nitrates, there was significantly less lactate production. This was significant in, let's say, 4 of the isolates and a trend in 1, and this was reaching a trend, but the pattern was clear that lactate appeared to be consumed. Then we looked at the pH as well. So here again, we have the control condition and the nitrate condition, and after 7 hours, the pH dropped more in the control condition than in the nitrate condition. And this also happened when comparing the control condition with the condition with isolate and nitrate, which we call a symbiotic condition. And what was very interesting is that one of the isolates prevented the pH drop more than just nitrate. So we think that this is an interesting probiotic candidate to prevent caries, and we would like to test this in the future. All right, that brings me to my conclusions. So we observed that nitrate can lead to health-associated changes in the oral microbiota composition and activity. So it can potentially decrease dysbiosis or increase eubiosis, which is an increase in health-associated species and function. And nitrate increases resilience against acidification when sugars are fermented. Rothia species can increase nitric reduction and lactic acid consumption capacities of oral communities. So they're interesting probiotic candidates. So you could also potentially increase eubiosis with these Rothia isolates. And in the future, we would like to check the effect of longer periods of nitrate supplementation, look at metatranscriptomic changes directly in dental plaque instead of saliva, look at species level changes, look at the effect on periodontitis and halitosis, and also test the probiotic candidates in vivo, looking at oral effects and systemic effects, and looking at their effects on the oral microbiota composition. I have 2 more slides. Would that be okay, Kiel, or is my time finished? All right. Because we published a preprint last month where we compared the tongue metatranscriptome of individuals with or without halitosis. And the colored boxes here are genes that were overrepresented in individuals without halitosis. And it is clear that nitrate reduction seemed to be, there were more nitrate reduction transcript in individuals without halitosis. So that further supports our hypothesis that nitrate reduction could be important to prevent bad breath. And by taking the genes that we found in this study together with our other study and a pioneering study that was done in 2010 that looked at denitrification genes in dental plaque. And the blue boxes are the genes that we found in our isolates, and the yellow boxes, the genes that we found in the metatranscriptome. We could expand the genetic overview or the potential genetic potential that is known so far. And we know now that the oral communities have the capacity to denitrify and to perform DNRA and nitrate assimilation from nitrite to ammonia. We even found some detoxification genes from nitric oxides to nitrites, and quite surprisingly also a gene involved in the fixation of nitrogen to ammonium. I think that a lot more remains to be discovered, and I hope to expand this map in future research. So yeah, with that being said, I've reached the end of my presentation. And I would like to give a special thanks to my PI, Alex Mira, and also Alejandro Artacho, a bioinformatician that helped with complicated analysis, and the rest of the oral microbiome lab, as well as the sequencing and bioinformatics department of FISABIO and the Spanish government for their funding. So here's a group picture before the pandemic, and that illustrates well the dynamics we have. We have a very positive and nice atmosphere. However, the group meetings nowadays look like this. That may sound familiar. We meet each other on Zoom every week, and hopefully after this winter, we can see each other in person again. So thank you very much for your presence, and I would like to receive any questions now, and also to propose to stay connected on social media. So yeah, you can also ask me any questions by email or on social media. Thank you.
Thank you so much, Bob. That was a fantastic tour de force through the oral cavity, which is obviously a fascinating territory to study, and many lessons you learned. These are always, of course, of high interest to us. And before I ask the participants here of this seminar here to raise their voices to ask questions, I think for me there's one comment and a specific question. The comment is very important and still a bit surprising for many people in the field, is the systemic effect we talk about. You talk about a specific area in the body, you talk about a very isolated part, the oral cavity, but your findings from other labs, as you have shown in these papers, that changes in the oral cavity, microbiome, metabolome, etc., can via the saliva go in the blood, and from the blood in the brain. This is not only frightening, but also, of course, just tells you that everything is connected to everything. And it also tells you, when you start from the other side, if you look into the brain and the changes and the Alzheimer patients, etc., you have to think systemically. You have to think holistically to understand what's really going on, because otherwise you will never get to the base of the disease. So thank you so much for that. I looked for some keywords which I'm missing in your talk, and that was colonization resistance, but you showed that in the first slide, because a rich microbiome prevents the intruders. But more important, my question is, how do the changes which you introduce, and nitrate and pH changes, how specific are they? You show changes in the composition of the microbiome, and certain species react and others do not react. So what's the mechanistic basis behind that? This is metabolic needs of this bacteria. And the second and more interesting question is, it's not only the composition which is changed, but I assume, we always talk, and you talk about the biofilm, but what we really should talk about is the biogeographical landscape. These microbes are not evenly distributed or randomly distributed in the oral cavity. They have, as other people have shown, beautiful spatial temporal organization and structure. Is that affected as well if you look at diseased oral cavities versus normal ones? I stop here, wait for your answer, and then I ask for questions from the audience.
Thank you, Thomas. I think that the summary you gave at the beginning was very accurate, and I agree that this holistic approach should be our aim for the future. And let's see. So yeah, of course, associations are not, causalities, but some things that are studied by other groups and are known that could explain the changes in the bacterial genera that we observed is that these are anaerobes and they're I briefly mentioned that, but they are sensitive against oxidative stress. So they incubated these bacteria in other studies with either metal oxides or nitric oxide releasing materials, and they were shown to be very sensitive against these molecules. Okay. So that's why we think that nitric oxide has a stronger effect on these bacteria than on other bacteria. But we are also working on different in vitro experiments to further support this idea. Mm-hmm. And what's also known is from in vitro studies already from decades ago is that Streptococcus, Veillonella, Actinomyces, and Oribacterium, they have representatives that really like sugar fermentation, and not because only sugar is their a nutrient, not only because of that, but also because when they decrease the pH, they can keep growing well, while other bacteria, like their neighbors, they cannot. So that's why we think that this has to do with the pH.
Mm-hmm. All right.
And also the structure of the biofilm, of course. Yeah. Those beautiful images that came out recently, they're very impressive. And we haven't looked at that yet, but this is something that we would absolutely love to do in the future. Yeah. So I hope to be able to give more details about that in some time.
I would really like to thank Bob Rosier. That was a fantastic tour in for us, kind of a not very common field of exploration. You showed us many promising new fields, and where you are going, we wish you the very, very best. We hope to see you here in person at some point. And thank you so much for the time and for sharing your very brand new results with us. Thank you so much, Bob.
Thank you for inviting me. It was a real pleasure.
Thank you.